Yes, ma'am.
Okay, suppose the next week Tuesday there is like again blockade, then is it possible for us to have the exam on Thursday, if Thursday is okay? Because at this moment we are not sure what is going to happen on Tuesday. If it is free, obviously, then we will have the exam on Tuesday, 19th. But if it is not, we have to keep like open the option. Maybe we can sit for the exam on Thursday. What do you say? Do you understand? I want to hear your opinion about this.
থার্সডে মানে কি ম্যাম 21 তারিখ?
Yes, because we don't know like next Tuesday if it is like a normal like day, or maybe there is blockade. We are not sure at this moment. Is not it?
Yes, ma'am.
So we should keep it open, whether it is Tuesday or Thursday. Okay, but you have to prepare yourself that the exam is on Tuesday. In case it is cancelled, then we will have it on Thursday. Hopefully one of the day should be fine.
Okay, ma'am.
Okay, everyone.
Okay, ma'am.
Okay, so this week Thursday, I hope we do you want to do the class this week?
No, ma'am.
No, ma'am.
Okay, so if you don't want
Friday
it,
exam, ma'am.
then it is fine with me. Okay.
Okay, ma'am.
Okay, so let us start with our lecture, today's lecture. Okay, can you see the slide?
Yes, ma'am.
Okay, so as I said that this lecture is important because up to now we have learned the two phases of metabolism, catabolism and the anabolism. Here we are going to focus on the integration of metabolism. That means how does the living organism actually coordinate and integrate the two phases of metabolism, especially because we are learning the metabolism of mammals, that mean human. So regarding that, we are focusing on this. And we actually already know we are multicellular organisms, and the cells, they are differentiated. That means there are different types of cells, and so the cells, they have different functions to do. Okay, so that function and the type actually require different fuel. That means the fuel requirement differs from one type of cell to the other, or we can say the particular tissue or organ as well. Okay, their energy requirement is different, as well as the pattern of the metabolism is different. Okay, so this integration and the coordination of the different metabolic activities of these different tissues and organs of the multicellular organism, how it is done, that is actually done by the hormones. So hormones are the one which is actually coordinating and integrating the various functions, metabolic functions, as well as the energy requirements of the different tissues and organs of our body. So that we are going to learn today. That means hormones. And as I said that actually in another course, GEB 203, where I think some of you, if you have already completed it, they are actually more um, uh, elaboration about the hormones. and that actually known as the endocrine system of our body, where all the hormones are discussed, what are their functions, what are what is their mechanism of action, everything is discussed there elaborately. And here we are actually focusing on the metabolism part. So if you look at the hormones, hormones are the one which is actually doing numerous control of different functions of our body, like starting from the maintenance of blood pressure, blood volume, electrolyte balance, embryogenesis, sexual differentiation, development, reproduction, hunger, eating behavior, digestion, and fuel allocation. Everything is actually coordinated by the hormones. Okay, the important system, actually how we communicate, or the different physiological functions happening within our body, that is done by the important system which we call neuroendocrine system. Actually, there are two different systems. One is known as the nervous system, another one is the endocrine system. Again, both are discussed in the physiology course, but here we are very briefly focusing in that topic. So these two system, they have some similarity, because some of the molecules, they function in both the systems. So together we call that neuroendocrine system. So in the neuronal signaling, actually in the nervous system, we can say the cells are called neuron. And the neuron, they have a different morphology. They have a cell like structure, and there is an extension, long extension from that circular structure. And their main function is to generate electrical signals, which is also known as nerve impulses, and those are generated in the cell body part. And then that signal travels through the long axon, that is the extension, and eventually it reaches the axon tip. And actually, when it reaches the axon tip, that causes the release of some molecules which are known as neurotransmitters. So those neurotransmitters are released from the axon tip, and they are these neurotransmitters, they actually diffuse within that intercellular or interstitial space, and then they bind to the receptors present on the target cell. And that target cell, that can be different types of cells, such as that can be another neuron, or another type of cell such as myocyte, or a secretory cell. And those target cells are actually only present, such as only a micrometer or few micrometer away site from the site of neurotransmitter release. So they are very closely associated with each other. Okay, so that is the way how the nervous system is functioning, and the nervous system actually releasing the neurotransmitter because of the electrical signals that causing the release. And then after it is released, it binds to the receptors present on the target cells, and the target cells are present very nearby with the actual cells which is releasing the neurotransmitters. Whereas in the endocrine system, hormones here, the molecules which are released here, we call hormones, and in the previous one, those are called neurotransmitters. So here hormones are released into the bloodstream, and then the blood is carrying the hormone, and then it is interacting with the target cells, and those target cells may be a meter or more away from the secreting cell. So that means in this case, hormones need to travel more distance if we compare with the neurotransmitters. Okay, so both the neurotransmitters and hormones, They actually how they like interact with the target cell. At first they will travel to that particular site where the target cells are present, and then they will interact with the receptors present on the surface of the cell. After that interaction, the cell will get the like a particular signal or signaling message what they need to do. Examples of like neurotransmitters as well as hormones. So there are some similarity. A single molecule can function as neurotransmitter or hormone. Epinephrine and norepinephrine. So these two, they can function as neurotransmitters. Where they are functioning as neurotransmitters? At synapses. Synapses is um, a space in between two neurons. Okay, two neurons are connected at synapses. So when they are released at that synapses, they are called neurotransmitters. And that synapses are very common in the brain, because brain is made up of the neurons. Or even neurons can be connected to muscle cell, that we called neuromuscular junction. And the neurotransmitters are released there and bind to the muscle cells. Or they can also be released function as hormones, where they bind to the target cells, for example liver cells or the muscle cells, and they regulate the metabolism of the cell. Okay, so that is their way of interaction, whether they are functioning as neurotransmitter or the hormone. Okay, next one, because the thing is that the hormones, because here we are not actually going to learn about the neurotransmitters. Rather, we will focus on the hormone action. That means how the hormone actually functioning, where it binds with the receptor and what happens to the cell. So we already come to know hormones bind to the receptors present on the target cell. And these receptors, they are like or we can say different type of cells, they have receptors for the same hormone. Okay, but the types of cells differ, but they are interacting with the same hormone. Now after the interaction, the function of the cell actually differ, because the cell is different about their activity. They are doing different functions, but those functions are done by interacting with the same hormone. Okay, so that means a single hormone can have different target cells, and those cells carry out different functions. And another important thing, that there is a specificity, or we say the structural complementarity between the hormone and its receptor. Like we learned that the enzyme substrate interaction, that is very specific. And here as well, the hormone is very specific for its receptor. And this specificity, which actually allows the high affinity of that interaction, and that is that will be done if very low concentration of hormone is present. That means if the hormone already occupied the bind the receptors, then the cell will get the signal. That is enough. So you don't need high concentration of hormone to bind to the receptor. Okay. Okay. Now another one. Where are these receptors present? We said that the cells, target cells, they have the receptors, and the receptors actually can be present on different locations of the cell, such as some of the receptors are present extracellular, that means outer surface of the plasma membrane. Some of the receptors are present within the cytosol, whereas others are present within the nucleus. So different hormones bind to the different receptors. It is not that a single hormone can bind all the receptors. It is the type of the hormone. According to that, that hormone will bind within the membrane or the cytosol or the nucleus. Now, the hormones which bind to the receptors present on the plasma membrane, that type of interaction actually triggers very rapid physiological or biochemical responses. So we will look at that, how that happens actually. Okay, now after the hormone receptor interaction, can you hear me?
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
Okay, so after the hormone interacts with the receptor, what happens within the cell? So there are uh, minimum six types of changes happening within the cell. Number one, the generation of a second messenger. And for example, we can say the cyclic AMP or inositol triphosphate. So in this case, the cyclic AMP is actually functioning as the second messenger, and that sometimes functioning as the allosteric regulator of one or more enzymes. So there are enzymes within the cell, and when the cyclic AMP is generated, that actually regulate the activity of those hormones by allosteric regulation. And another one, a tyrosine kinase is activated. That means tyrosine kinase activity is associated with the receptor, and the hormone receptor interaction activate the tyrosine kinase activity. And then tyrosine kinase, we know it is the enzyme which adds the phosphate group to the substrate. So here as well actually, maybe the protein which is phosphorylated, that becomes activated or inactivated. Then another second messenger, which is cyclic GMP, that is also activated by the enzyme guanylyl cyclase. That means guanylyl cyclase is attached with the receptor. And then another one, a change in membrane potential after the interaction of hormone and receptor. How that happen? The changes, because when the hormone binds to the receptor, that causes the opening or closing of a membrane, sorry, hormone gated ion channel. That means hormone gated means when the hormone binds to the receptor, because of that interaction, a particular ion channel will be open or closed. And when it is open, obviously there will be movement of the ions inside the cell, or maybe the ions are coming out of the cell, and that causes changes in the membrane potential. Then they function like as the adhesion receptor. That means they are interacting with molecules present in the extracellular matrix, and after that interaction, the information is delivered to the cytoskeleton, that means interior of the cell. And other one, the steroid, or like a steroid like molecule. That means some of the hormones, their nature is like they are steroid hormone, and these hormones, they eventually actually changes the expression of certain genes. That means they can interfere with the transcription as well as translation, and these hormones usually they bind to the receptor which are present within the nucleus. So they function as a complex, hormone receptor complex, and interact with the regulatory sequences of their target gene. So these are the major six changes occurred within the cell after the hormone interacts with its receptor. Okay, next one, that is about the nature of the hormones, or we can also say classification of hormones, which can be done in several ways. The first one, that is about the chemical nature of the hormone. So what is the composition of the hormone? According to that, the hormones can be classified. So the first one is, that is the makeup of the hormone. So hormone can be made up of different molecules. Hormone can be made up of peptide. That means hormones are protein in nature. Example, insulin and glucagon. And how these hormones are synthesized by the proteolytic processing of prohormone. That means at first hormones are released as inactive molecule, and then those undergoes proteolytic degradation and then they become active. Then catecholamine, they are made up of sorry, example is epinephrine, and they are synthesized from the amino acid tyrosine. Then another hormone, icosanoid, prostaglandin, example, and they are synthesized from the fatty acid arachidonate or arachidonic acid. So, and these hormones, if you look at that, how are they functioning? These hormones, they are interact with the receptors which are present on the plasma membrane, and when they interact with the receptors within the cell, there is the generation of the second messenger. That is their mode of action. Okay, now another hormone, steroid hormones. In the previous, I think during the lipid biosynthesis, we learned that steroid hormones. Example is the testosterone, and they are synthesized from cholesterol. Then vitamin D is actually used to synthesize hormone, and the particular name of the hormone is 125 dihydroxy cholecalciferol. That is also synthesized from cholesterol. Another hormone is retinoid. Example is the retinoic acid that is synthesized from vitamin A. Another one, thyroid hormone, and example is the triiodothyronine, which is synthesized from tyrosine, amino acid tyrosine, and that is actually happened within the thyroglobulin of the thyroid glands cells. And these hormones, this group of hormones, which are actually mainly steroid nature, that means they are lipid um, molecule, and their receptors are present within the nucleus. So when they bind, they actually like traveled to the nucleus of the cell, bind to the receptors, make the complex hormone receptor complex, and then this complex interact with the target genes regulatory sequences, which ultimately regulate the transcription of the gene. Okay, so these are the hormones. They are they do their function in this way. And another hormone in this table, that is the nitric oxide. Nitric oxide itself is the hormone, and that is synthesized from the amino acid arginine, and arginine is actually oxidized, and then the hormone is synthesized. And this hormone that has the receptor present within the cytosol. Okay, within the cytosol the receptor is present, and this receptor ultimately activate the enzyme guanylyl cyclase, and that produces the second messenger cyclic GMP. Then cyclic GMP does its function. Okay, so this is the one way of classification according to the nature of the hormone, and there is other ways. And here the second way, where the hormones are classified from where they are released and where are their target tissues are present. According to that, there are three types of hormones. The first one is called endocrine, and it is coming from the Greek words. Endon means within, crinin means released. So here the glands, which actually we call the endocrine gland, the hormones are released from the endocrine glands into the blood, and then the blood carries the hormone to the target cells. Okay, target cells can be anywhere in our body, and they bind to the target cells and do their function. Example, insulin and glucagon. Insulin and glucagon, they are released by the pancreatic or pancreatic beta island cells, and then they actually have receptors present on all the cells of our body. Second type, paracrine hormones. Here the hormones are released into the extracellular space. Okay, so That means they are not secreted into the blood, rather they are secreted into the extracellular space, and where that hormone is diffused and diffused to the neighboring cells. The neighboring cells, they have the receptors, and the hormones interact with it and do their function. Example, icosanoid. And the third one is autocrine. Actually here the hormones which is secreted or released by the same cell has the receptor for the hormone. So the hormone interacts with its own receptor and do the function. Example of such hormones is cytokine. Cytokine has very important role in our immune system. Okay, so now if you look at the mechanism of the action, so in the previous one as well, actually at the very first type of classification, there we have also focused on the mode of action. Okay, some of the hormones, they bind to the receptors present on the plasma membrane and they generate second messenger, whereas other hormones, their receptors are present within the nucleus, and then the hormone receptor complex actually regulating the transcription of a gene. So actually that is the thing here. It is discussed more elaborately, and we can say the hormones actually have two mechanisms of action, and that is also actually done based on the nature of the hormone. And those hormones which are water soluble, such as peptide and amine hormones, example insulin is the peptide hormone and amine hormone is the epinephrine. So they are water soluble, so they actually cannot cross the plasma membrane because plasma membrane is hydrophobic. So these kind of hormones, they cannot cross the plasma membrane, so they bind to the receptors present on the plasma membrane. And after this interaction, actually the extracellular domain where the hormone binds, the receptor undergoes a conformational change. So the same thing we have learned in case of like interaction of the hormone, enzyme with the substrate, or even like ligand binding to the protein. So after that interaction, we know that the protein undergoes conformational change, or even we have seen the same mechanism for allosteric protein. There is conformational change. And this conformational change actually is the one which is triggering the downstream effects of the hormone. Now, if you look at the figure where you can see the peptide or amine hormones binding to the receptors present on the plasma membrane, which generates the second messenger, for example, cyclic AMP. And the cyclic AMP is actually allosterically regulating the pre-existing enzyme present within the cytosol. The enzyme is already present, maybe it is not active yet. After the allosteric modification, it will be activated. Okay, and that incidence takes just few seconds to happen, whereas in case of water insoluble hormones, examples steroid or thyroid hormones, they can easily pass the plasma membrane because they are also hydrophobic in nature. They can easily cross the plasma membrane, which is also hydrophobic, and then eventually they can cross the nuclear membrane and go inside the nucleus where the receptor is present. They interact with the receptor, make the hormone receptor complex, and then this complex interacts with the regulatory sequences of a target gene. And this interaction actually influences the transcription of that gene. So maybe the transcription rate has been increased, or it can be decreased, or even the gene which was not previously transcribed within the cell, after this interaction the gene is transcribed. Okay, so that is the function or the mechanism of this group of hormones. Is it clear?
Yes, ma'am.
Okay. Now, actually, up to this point, we have focused that hormone receptor interaction doing some catalytic activities within the cell. That means it is functioning as a catalyst. Okay. Now, here, this hormone receptor interaction not only functioning as the catalyst, this is also actually functioning as a signal transducer or signal amplifier. So we will understand this by the like here you can see the uh, breakdown of glycogen, which is actually like the cell receives the signal by the interaction of the hormone epinephrine. So the situation here, what is that? We can say when the organism they don't have enough glucose. So in that situation, obviously the command will come from the central nervous system, and then it will cause the release of the hormone epinephrine. So epinephrine is actually released from the gland adrenal medulla. So here if you look at this figure, epinephrine is released, like X molecules of epinephrine is released, and obviously so X molecule will bind to X molecules of receptor present on the target cell. Here the target cell is hepatocyte, the liver cell. Okay, because that is very specific. It is not that you need 2x to bind to the X receptors. So that is very specific, we already learned, and only that minimum amount of hormone is enough to interact with the receptor. Okay, after that, in this case, what is happening? We already come to know how this hormone is functioning. Epinephrine is the amine hormone, and that bind to the receptors present on the plasma membrane, and that interaction eventually generates the second messenger. And here you can see how the second messenger is synthesized. At first, actually the hormone receptor interaction activates a protein which is called G protein, that is just present in our surface of the plasma membrane, and that activates the protein or enzyme adenylyl cyclase, which then synthesized cyclic AMP from ATP. And here already you can see the signal like transduction or transduce, or the signal is amplified. So X molecules of epinephrine generates 20 X molecules of cyclic AMP. And we already know cyclic AMP is functioning as the second messenger, which is responsible for allosteric modification of the target enzyme. And here it is protein kinase A. So once the protein kinase A is activated, because under the influence of the cyclic AMP, we can see 10x molecules of protein kinase A is activated, which now phosphorylate the target protein here, and its target protein is phosphorylase B kinase, and 100x molecules of phosphorylase B kinase is activated, and this in return activate its target protein or enzyme, which is the glycogen phosphorylase B. So glycogen phosphorylase, we have learned it, if you remember, during the how the endogenous glycogen is releasing glucose, and the glucose is entering the glycolysis, if you remember. So ultimately we get 1000 X molecules of active glycogen phosphorylase A, and that is the enzyme which is working on the substrate, that is glycogen. Glycogen is the storage form of glucose within hepatocyte, and the glycogen will be broken down as glucose 1 phosphate, and 10,000 X molecules of glucose 1 phosphate is released from the glycogen by the action of glycogen phosphorylase A. And after that we get glucose from glucose 1 phosphate by several steps, and eventually the glucose will be released into the blood. So 10x molecules of glucose is released because of X molecule of epinephrine interacting with the receptors of the target cell. So that is the signal amplification. Is it clear? Do we
Yes, ma'am.
understand? So in this same like figure, two things we are seeing: transduction of the signal, how the signal is transduced from one step to another, and also the amplification of the signal. Okay, now again, this is actually about the uh, we can say mechanism of action that actually lead to the response time of the target cell, or the hormone's action. We already know hormones, they bind to the receptors, whether the receptors are present on the plasma membrane or they can be present within the cell, specifically within the nucleus. So the hormones which are interacting with the receptors present on plasma membrane, in that case the response is very rapid. That means the physiological or biochemical responses, that is very rapid, and that only takes seconds. And here the example is the this is the one which is happening, that actually that means starts from the secretion of the hormone, which is the epinephrine, that is secreted from the adrenal medulla, and that is secreted into the bloodstream. And epinephrines, here the target cell is skeletal muscle. Okay, we know that glycogen is also stored within the muscle cell, and the result is release of glucose from the glycogen. Okay, and that just takes seconds. Okay, seconds. Why? How is that possible? Because in this case, these hormones, they are actually functioning in this way, that already they are substrates present within the cell, just they need to be activated. Okay, whether by allosteric modification or phosphorylation, they are activated and then they can do their function. বুঝতে পেরেছি আমরা এটুকু?
Yes,
Yes,
ma'am.
ma'am.
So why
Yes,
it
ma'am.
is happening so fast? Because they are already there, their targets are already there, just they are in inactive state. Just by the allosteric modification or covalent modification, means phosphorylation, they are becoming activated and then they can do the work. Whereas the thyroid hormones and the sex hormones, the steroid hormones, they need like long time to get the response, because in that case they have to travel to the nucleus, they bind with the receptor, and the complex hormone receptor complex interact with the regulatory sequence of the target gene, and then they do the transcription, or maybe they are upregulating the transcription or downregulating the transcription, or maybe the gene which was not expressed earlier, that will be expressed. So that takes time. So transcription will happen, then the mRNA will go to the cytoplasm where the translation will happen, and then again we know that after the translation there are post translational modifications, and after that the enzyme or the protein will be activated, then it can do the action. So that takes time. So there is like maximal response, so takes longer time, which is like even hours or days. Okay, now you look at the regulation of hormone release. That means when the hormone is released and when the hormone release is turned off. Obviously, that release, actually specifically the release is done by the central nervous system, and central nervous system, that is receiving all the informations within our body as well as from the environment, and the information can be in any form, such as infection, fear, pain, hemorrhage, hypoglycemia. So all these informations will be gathered by the central nervous system, and the central nervous system then it will command the respective organ to carry out the action to control the situation. Okay, so in this case, particular example here, it is shown uh, the expression or the release of the hormone cortisol, which is released from the adrenal gland. And specifically, this is done because of the hypoglycemic situation. That means when the glucose level, blood glucose level is low. So if that situation arises, the central nervous system signals the hypothalamus, that is also part of our brain. Hypothalamus then release the hormone which is called corticotropin releasing hormone. In this slide also shown the amplification. So that corticotropin releasing hormone is released in nanogram. These hormone then induced another part of the brain known as the anterior pituitary. Anterior pituitary is also function as the endocrine gland or part of the endocrine system. Anterior pituitary then releases adrenocorticotropic hormone, and that is released as microgram. Now, this adrenocorticotropic hormone directly binds to the target organ, which is the adrenal gland. And adrenal gland is the hormone which actually release the ultimate hormone, which will functioning in this situation. That means the hypoglycemic situation will be controlled by the cortisol. So the cortisol is released in milligram, and they will then bind to the target tissues, that means or cells, muscle cell, liver cell, adipose tissue. Okay, that is their target. And when, what will be the result? Can anyone, can we guess it? Cortisol here targeting muscle, liver and adipose. If the situation is hypoglycemic, what will be the result? কর্টিসল কি করছে এখানে? কেউ কি আমরা বলতে পারবো? হুম, কেউ কি আমরা চিন্তা করতে পারি এখানে কর্টিসল যেই তার টার্গেট সেলগুলোকে বাইন্ড করছে, তারপর টার্গেট সেলগুলো কি করছে? হ্যালো, আমরা কি চিন্তা করতে পারি? Can you think about that? Already I said the situation is hypoglycemic. That means low blood sugar. তাহলে কি হচ্ছে এখানে? এই হরমোনটা কি করছে? এই টার্গেট অর্গানগুলোকে সে যে বাইন্ড করলো, তারপর সেই টার্গেট অর্গানগুলো ওখানে কি হচ্ছে? What is the main purpose of this hormone? Hmm?
Glucose synthesis.
Yes, because the situation is hypoglycemic, and the that means we have to get back to the normal blood sugar is required. So cortisol targeting the cells or the tissues to, like, the first one obviously is to synthesize glucose and release it into the circulation. Okay, now when the job is done, so that means the glucose level returns to normal. তখন কি হবে? There is no need of cortisol anymore. So this release of cortisol should be turned off. কিন্তু সেটা কিভাবে হবে? এখানেই বলে দেওয়া হচ্ছে, that is happening by the hormone itself. So cortisol is actually turning off its own release. How? By inhibiting the previous inhibiting the release of previous hormones in the pathway, যেটাকে আমরা বলি feedback inhibition. Okay, so cortisol is inhibiting the hypothalamus as well as the anterior pituitary to release the hormone corticotropin releasing hormone and adrenocorticotropic releasing hormone respectively. Okay, বুঝতে পারলাম?
Ji ma'am.
Ma'am,
Okay, next one, what We said at the very first that we are multicellular organism and the cells are differentiated, as well as there is that different fuel requirement by the different tissues and organs of our body. So in this slide, it is shown that what are the different functions of the important organs or tissues of our body. So if you look at this slide, in pancreas, if you look at the pancreas, or we can start with the brain. Brain is the one which is actually part of our central nervous system. That is actually one which is receiving all the information and then commanding all the organs to control the situation, every situations actually. And how the brain is doing that? By the generation of membrane potential. Okay, so to maintain the membrane potentials, the brain continuously need the supply of fuel. Okay, and we already know that is actually the excellent fuel is glucose. But now we already come to know if glucose is not available, brain has the ability to use another form of fuel, which is ketone bodies. Okay, now pancreas is actually uh, here the endocrine function of the pancreas is shown. Pancreas secretes the two hormones, insulin and glucagon, which has important role to maintain the blood glucose level. Then liver is the one, or it is actually known as the chemical factory, which actually receives all the nutrients. Those are like uh, absorbed in our digestive system after digestion of the foods, that is the glucose, amino acid, fatty acid, all these will go to the liver. And liver is the one which is responsible for the distribution of the fuels to the other organs of our body. Okay, portal vein is actually that is the like a transport system from our digestive system to the liver. So through the portal vein, all the nutrients travel from the intestine to the liver. Small intestine is the part of our digestive system which is responsible in the digestion as well as absorption of the nutrients, and then from the small intestine all the nutrients goes to the blood circulation. Lymphatic system's main job is to carry the lipids, that means from the intestine to the liver, and that is done by the lymphatic system. And adipose tissue, already we have learned about that. Their main function is to store triacylglycerol. Whenever necessary, triacylglycerol will be degraded and fatty acids will be released, which can be utilized to synthesize the fuel. And the muscle is the one which actually need the energy, that is ATP, for the contraction, and it can do it by the breakdown of glycogen, or the if that is not enough, that can be transported from liver. So liver is the one which is getting all the nutrients through the portal vein, and then liver actually distributed the nutrients to other organs of our body. And in the liver, there are two types of cells. One is called Kupffer cells. They are actually phagocytes. That means their function regarding whether there is any foreign substance or pathogen is present. That is their job. That means they are protecting our liver. And another one is the hepatocytes are the one which is actually doing all the metabolic reactions. Okay, so look at the glucose metabolism in liver. If you remember, this is actually what we have learned about the glucose metabolism, whether it is the catabolism of glucose or the biosynthesis pathway. And glucose enters the liver by the transporter, which is called glucose transport exercise or other activities. That means more energy is required at that moment. It breaks down the glycogen, muscle glycogen, and that undergo fermentation, and glucose is fermented into lactate. Okay, so during that vigorous activities, or another source they can also use during such activity, that is phosphocreatine. So phosphocreatine is another phosphoryl potential compound which has the ability to deliver the phosphate group to its substrate. So by that ATP is produced, and that ATP is used during the muscle contraction. Okay, now another important function, actually we have learned it before, that is known as Cori cycle, which that is about the metabolic cooperation between two different organs or tissues, we can say. So during the exercise, where the glycogen is broken down and glucose is fermented to lactate, that lactate is actually transported to the liver through the blood, and the lactate is um, used to synthesize glucose by gluconeogenesis, and this glucose then transported back to the muscle to be stored as glycogen, so that whenever it is needed, the glycogen can be broken down and energy can be produced. So that is known as the Cori cycle. So that means two organs or two different tissues, they are cooperating to fulfill the requirement of energy. Okay, now the brain. Brain, we know already, that is the one which actually requires the supply of fuel constantly, even during the starvation. Already we learned during such situations, liver is the one which is synthesizing ketone bodies from fatty acids and supplying that to the brain so that brain can utilize it. And actually in the normal situation, when blood glucose level is normal, so brain can utilize glucose to generate the energy. But when there is no blood glucose, that means it declines, then the brain can utilize ketone bodies. So here they need the energy ATP, because as I already said, to maintain the membrane potential, and that is done by the particular protein known as the sodium potassium ATPase, and that maintains the membrane potential, and ATP is used by this enzyme or the transporter sodium potassium ATPase. Okay, so next one, we will come back to the hormone again, hormonal regulation of fuel metabolism. In in a healthy person, we know that the blood glucose level, which is about 4.5 millimolar. So how this uh, blood sugar level is kept in that range? That is possible because of the actions of the hormones. Not only one hormone, several hormones are working to keep the blood glucose level about 4.5 millimolar. And the hormones are insulin, glucagon, epinephrine, and cortisol. All these hormones, they actually work on different tissues. Specifically, we can say liver, muscle, and adipose tissue. The thing is that when the blood sugar level is high, at that time, the insulin will signal all the cells of our body so that the excess glucose is taken by the cells, and then the cell will utilize it. Specifically, when there is enough glucose, they will convert it to glycogen or triacylglycerols for storage. So at that stage, their main job is to store the fuel as glycogen or triacylglycerol. But another situation, if the blood glucose level is low, then the signal is provided by the glucagon, another hormone. And in that case, the tissues, they get the signal that blood sugar level is low. So what they need to do, they have to synthesize glucose and release that glucose to the blood so that the sugar level comes to normal. And That job is specifically done by the liver by gluconeogenesis, or maybe at that stage the cells they can oxidize fats because the glucose level is low. Another hormone, epinephrine, when the blood glucose level is low, epinephrine is released, and epinephrine then targets the muscles, lungs, and hearts, so that the glucose synthesis or the stored carbohydrates can be degraded, and the cell can do the functions which is necessary at that moment, because that is not a normal situation. And finally, cortisol actually also responsible for body's response when we are facing long term stresses. In that case as well, the thing is that the cells must maintain normal functions, and during that situation the blood glucose level should be maintained at a normal range, otherwise the cells cannot perform properly. So the first one is the insulin. So we already know that when the blood sugar level is very high, because after a meal, so after a meal we have consumed carbohydrate, protein, or fat. So all those are digested in our digestive system, and the blood sugar level goes up. In that situation, the signal is given to the pancreas, and pancreas releases insulin, and insulin in return signals the tissues, specifically we can think of liver. So then the liver, what it does, obviously because the blood sugar level goes up, so it will intake the glucose to keep the blood sugar level normal. And what the liver is doing here, so the glucose inside the liver, that can be catabolized by the normal process we have learned, or it can be stored as glycogen, or the fats which are we are taking, the fats will be absorbed, and through the lymphatic system they will go to the adipose tissue to be stored, or maybe muscle cells can utilize it to generate energy, or even the excess glucose ultimately they will be, so like at first we will get the acetyl CoA, and acetyl CoA is the precursor of the fatty acid, from where the triacyl glycerol will be synthesized, and then the triacyl glycerol can be transported to the adipose tissue or the muscle through the VLDL, which is the very low density lipoprotein. Their function is to carry or transport the triacyl glycerol to the different organs of our body. Okay, or the amino acids, those again from the our intestine, it goes to the liver through the circulatory system, blood, and then the amino acids, they can be catabolized, or they can be utilized to synthesize the protein. So in this state, if you look at here, what is the ultimate result or metabolic state of the liver? Because there is enough glucose. After you utilize glucose as the fuel, so what will happen to the rest of the glucose? The excess glucose is ultimately converted into fat for store. Okay, so we say that in well fed state, that means when we have food or meal, after that the metabolic state of our liver is lipogenic. The liver is mainly converting the excess glucose into fat, and that fat is ultimately stored within adipose tissue. Obviously before that it should fulfill the energy requirement of all the organs of our body. Is it clear?
Yes, ma'am.
Okay. We said that when the blood sugar level is high, then the pancreatic beta cells secrete insulin. So there is a mechanism for this and how it is done. So we can see here when the glucose level increased, glucose is transported into the beta pancreatic beta cells through the glucose transporter GLUT2, and in the cell glucose is oxidized by glycolysis, and eventually the TCA cycle, oxidative phosphorylation, ultimately we get ATP. That means ATP concentration increased, and this higher level of ATP then cause the closing of potassium channel. So potassium channel, that is also important for the membrane potential of the cell. When it is closed, that causes a transmembrane like differences within the cell, transmembrane like electrolytic differences in the cell, that is called VM, and we call that membrane is depolarized. Okay, depolarized. And because of that, now this depolarization is causing the opening of another channel, that is voltage gated calcium channel. So voltage is that electrical gradient. So when there is this changes, that causes the opening of the calcium channel, and through that calcium enters the cell. Eventually, that also at the same time within the cell, the sorry endoplasm yes endoplasmic reticulum, so they also store calcium, those will be released. So both these calcium release, one is coming from outside of the cell through the calcium channel, and another one is released from the stored, uh, they will increase the calcium concentration. And when the calcium concentration goes up, that will cause the exocytosis of the granules which are storing insulin. Okay, so then the insulin is released, and then insulin goes to the target cells, bind to the receptor, and signals the cell to take the glucose. বুঝতে পেরেছি আমরা?
Yes, ma'am.
Yes, ma'am. আচ্ছা, এখন আমার কোশ্চেন, এখানে যে ইনসুলিন গ্র্যানুলস, এটা কোথা থেকে আসলো? কেউ কি বলতে পারবে আমাকে? এখানে আমরা দেখছি নিউক্লিয়াস, তারপরে একটা পরে অরেঞ্জ গ্র্যানুলস দেখছি, দোজ আর দ্য নিউক্লিয়ার ইনসুলিন গ্র্যানুলস। বাট আমার কোশ্চেন কোথা থেকে আসলো সেলের ভিতরে, কিভাবে আসলো? কেউ কি বলতে পারবো? হুম, আমরা কি কেউ বলতে পারবো এখানে ইনসুলিন গ্র্যানুলস গুলো কিভাবে আসলো?
ম্যাম আয়ন ট্রান্সফারের মাধ্যমে।
হ্যাঁ, আয়ন ট্রান্সফারের মাধ্যমে মানে কি? ইনসুলিন ইনসুলিন কি একটা আয়ন? ইনসুলিন কি?
ও ম্যাম,
হোয়াট ইজ ইনসুলিন?
ম্যাম হরমোন
হরমোন
হরমোনটা
ম্যাম।
এখানে কিভাবে আসলো?
ব্লাডের মাধ্যমে আসছে ম্যাম,
হ্যাঁ,
ব্লাডের মাধ্যমে।
ব্লাডের মাধ্যমে, তার মানে ইনসুলিন কোথায় তৈরি হয়েছে? ব্লাডের মাধ্যমে আসছে বলছো, তাহলে কোথায় তৈরি হয়েছে ইনসুলিন? তুমি যে বললা ব্লাডের মাধ্যমে মাধ্যমে আসছে, তাহলে ইনসুলিন কোথায় তৈরি হয়েছে বলো? হুম, ইনসুলিন আরেক জায়গায় তৈরি হলো ব্লাডের মাধ্যমে, তারপরে এই সেলের ভিতরে ঢুকলো, এরকম কিছু? তো সেলের মধ্যে ঢুকে বসে থাকলো, তারপরে এটা রিলিজ হইলো, এরকম? হ্যাঁ, কিভাবে হলো বলো দেখি। 202 কয়জন করছো? হুম, অনেকে তো 202 করছো। যারা 202 করছো তারা তো এটা বলতে পারার কথা। ওখানে ইনসুলিন কেমনে আসলো? হ্যালো, আই এম আস্কিং সামথিং। কিভাবে আসলো বলো? সময় নাই তো বেশি, শেষ করতে হবে আজকে লেকচার। তাড়াতাড়ি বলো কিভাবে আসলো?
প্যানক্রিয়াস থেকে।
আমরা তো প্যানক্রিয়াসের ভিতরেই আছি। প্যানক্রিয়াসের ভিতরে প্যানক্রিয়েটিক বিটা সেলের মধ্যে আছি। তো প্যানক্রিয়াসের থেকে আসলো সেটা তো বলে কোনো লাভ নাই। আমি প্যানক্রিয়াসের ভিতরে প্যানক্রিয়েটিক বিটা সেলস এর ভিতরে দেখছি। এই সেলের ভিতরে আমার কোশ্চেন, এই সেলের ভিতরে এই যে ইনসুলিন গ্র্যানুলস গুলো কোথা থেকে কিভাবে আসলো? সেটা হলো আমার কোশ্চেন। বলো এখন। কয়জন করছো 202? হ্যাঁ? অনেকেই করছো। বলতে পারতেছো না কেন? কিভাবে পাইলাম আমরা ইনসুলিন? একটু মাথা খাটাও। কিছুই নাই মাথায়? কিছু মাথায় আছে নাকি নাই? মাথা যদি না খাটাও তাহলে এরকম সিচুয়েশন তুমি কিভাবে এক্সপ্লেন করবা? বলো, আজকে যদি ইনসুলিন কোথা থেকে আসলো না বলতে পারো তাহলে তো ফাইনাল এক্সামের একটা কোশ্চেন এমনি মাইনাস হয়ে যাবে। কিছুই শেখো নাই তাহলে। 202 কিছুই শেখো নাই এটা যদি বলতে না পারো। তা 202 আগে 201 এর আগে 202 করে লাভটা কি হইলো? হুম, বলো। 202 আগে করে লাভটা কি হইলো? এটার অ্যান্সার যদি করতে না পারো, কেন করতে গেছো 202 আগে? বলতেই যদি না পারো এখানে ইনসুলিন কোথা থেকে আসলো? ইনসুলিন কি একটা হরমোন, তাই না? নাকি না?
ইয়েস ম্যাম, হরমোন।
কেমন কেমন হরমোন? কি টাইপের হরমোন ইনসুলিন? ওর কেমিক্যাল কম্পোজিশন কি? আজকেই তো পড়লাম। ইনসুলিনের কেমিক্যাল কম্পোজিশন কি? ও মাই গড, ইনসুলিনের কেমিক্যাল কম্পোজিশন কি? ইউ হ্যাভ
ম্যাম
ইউর স্লাইড উইথ ইউ।
অ্যামাইনো অ্যাসিডস থাকে।
হ্যাঁ, ইট ইজ প্রোটিন ইন নেচার। তাহলে একটা প্রোটিন কিভাবে সেলের ভিতরে তৈরি হয়? একটা প্রোটিন কিভাবে সেলের ভিতরে তৈরি হয় বলো? বলেই তো দিলাম, বলো। সেল কিভাবে একটা প্রোটিন তৈরি করে? তাও বলতে পারে না। প্রোটিন কিভাবে তৈরি হয়? হ্যাঁ, একটা সেল কিভাবে একটা প্রোটিন তৈরি করে?
Ma'am, translation.
ম্যাম ট্রান্সলেশন।
শুধু ট্রান্সলেশন হলে প্রোটিন হয়ে যাবে? ট্রান্সলেশনের আগে কিছু
ট্রান্সক্রিপশন।
দরকার নাই? হ্যাঁ,
Ma'am, transcription.
হ্যাঁ। তো ট্রান্সক্রিপশনের আগে কি থাকতে হবে? কি ট্রান্সক্রিপ হবে?
এমআরএনএ।
এমআরএনএ ট্রান্সক্রিপ হবে নাকি ট্রান্সক্রিপশনের পরে আমরা এমআরএনএ পাবো?
Ma'am, DNA.
পরে
ডিএনএ।
পাবো ম্যাম।
ডিএনএ।
ট্রান্সলেশন হবে, তারপর এমআরএনএ পাবো।
ডিএনএ না বলে কি বলবো? সব ডিএনএ, ডিএনএ তো দ্য ক্রোমোজোমে এভরিথিং ইজ ডিএনএ। কি বলবো? কি ট্রান্সক্রাইব
Ma'am,
হয়?
ম্যাম জিন।
ma'am, ribosome.
হ্যাঁ, তো এত সময় লাগে কেন তোমাদের? তো তোমার সেলের ভিতরে কি ইনসুলিন জিন নাই নাকি আছে? হ্যাঁ, তোমার এই যে প্যানক্রিয়েটিক সেল, সেই সেলের জিনোম আছে, তাই না? সেই
ইয়েস
জিনোমের
ম্যাম।
মধ্যে ইনসুলিন জিন আছে
জি ম্যাম।
নাকি নাই? আকাশ থেকে পড়ছে ওখানে, না? আকাশ থেকে ওখানে ইনসুলিন এসে পড়ছে। তোমাদের ভাবসাব দেখে তো সেটাই মনে হয় আমার। এই প্যানক্রিয়েটিক সেল যেটা বিটা সেল পড়ছো তার তো একটা জিনোম আছে। সেই জিনোমে ইনসুলিন জিন আছে। সেই জিনটা ট্রান্সক্রাইব হচ্ছে, তারপরে ট্রান্সলেশন হচ্ছে, তারপরে ইনসুলিন প্রোটিন তৈরি হচ্ছে। That protein undergoes post-translational modification and finally it is stored within the granules, vesicles আমরা বলি। এখানে আমরা বলছি ultimately that is stored within the granules। সেখানে স্টোর অবস্থায় আছে এবং সেই স্টোর অবস্থায় থেকে সে রিলিজ হবে কিভাবে, that is happening by exocytosis process and how that happen সেলের ভিতরে যে কাজগুলো হচ্ছে সেটা এখন দেখলাম। তো কেন তোমরা এগুলো রিলেট করতে পারো না? কেন একটা কোর্সের সাথে আরেকটা কোর্সের কোনো লিঙ্কেজ তোমাদের মাঝে নাই কেন? এটা খুবই অবাক লাগে। তো 202 আগে যে করলা, তো তোমার 202 কি হেল্প করলো তোমারে এখানে? এটুকুই বলতে পারলা না। ওকে, সো that is the way insulin granules are here. Okay, now if we are not having a meal for hours, we are fasting, then what will be the situation? That will cause low blood sugar, and that low blood sugar will be signaled by the hormone glucagon, which is also released by the pancreas. Glucagon signals the liver, and then the liver's job is what? কি করবে লিভার তখন? ব্লাড সুগার লেভেল যদি কম হয়, বলো কি করবে? সব পড়ে আসছো, এখানে জাস্ট সামারি দেখতেছো। বলো লিভার কি করবে? ব্লাড সুগার লেভেল
ম্যাম
কম, লিভার কি করবে
গ্লুকোনিওজেনেসিস।
তখন?
ম্যাম গ্লুকোনিওজেনেসিস।
Yes, the first job is gluconeogenesis. It will produce glucose by gluconeogenesis. Gluconeogenesis কোথা থেকে হবে? নন কার্বোহাইড্রেট সোর্স থেকে হবে, তাই না? যেটা আমরা অলরেডি জানি, অ্যামাইনো অ্যাসিড, some of the amino acids are glucogenic. Those will be used to synthesize glucose by gluconeogenesis. Or আরেকটা সিচুয়েশন হতে পারে, fatty acids will be released by the degradation of triacyl glycerol, তাই না? Those will be then used as the fuel, সেটাও হতে পারে। অথবা লিভার আরেকটা কাজ করতে পারে, কিটোন বডি সিনথেসিস করতে পারে from the fatty acid degradation, and then ketone bodies will be transported to other organs. And so in this state, what can we say? What is the main purpose of the glucose liver? কি করছে গ্লুকোজ? মেইন পারপাসটা কি? বলো। গ্লুকোজ সিনথেসিস, তাই না? সেটাই তো মেইন পারপাস। তাহলে মেটাবলিক স্টেট অফ গ্লুকোজ in or during low blood glucose situation is glucogenic. Okay, গ্লুকোজেনিক। গ্লুকোজের মেইন জব এখানে কিভাবে গ্লুকোজ সিনথেসিস করবে, and that glucose will be released into the circulation as well as other organs may get the fuel. ওকে, এখন সিচুয়েশন যদি আরো খারাপ হয়, such as একটা তো বললাম যে fasting, and that fasting is prolonged, or uncontrolled diabetes mellitus, সেক্ষেত্রে কি হতে পারে? সেক্ষেত্রে হতে পারে প্রোটিন ডিগ্রেড হবে, protein will be catabolized, and we know amino group is released, that is further catabolized, and the nitrogenous waste product is produced. In our case it is urea, and amino acids that will be utilized to synthesize glucose by gluconeogenesis. And actually the situation is that here the intermediates of the TCA cycle are utilized to synthesize glucose. So when the intermediates are removed, the TCA cycle cannot go on. So that will eventually lead to the accumulation of acetyl CoA. When the acetyl CoA accumulates, কি হবে acetyl CoA we can get after the degradation of fatty acids, and that accumulation leads to the formation of ketone bodies, and then the ketone bodies will be supplied to the other organs to be utilized as fuel. বুঝতে পেরেছি?
Yes, ma'am.
এখানে এখানে কিন্তু মেইন যেটা আমরা দেখতে পাচ্ছি টিসিএ সাইকেলটা ইন্টারাপ্টেড হচ্ছে, because the removal of the intermediates which are used to synthesize glucose by gluconeogenesis. Okay, so the hormone epinephrine, which we have already seen, and the epinephrine is also actually like controlling the situation when the glucose level is low. And specifically, the situation arises in a stressful condition. So suddenly we are in a stressful condition, such as we have to fight, and what our cells will do. So obviously, the cells need to do the work, energy is required, and the energy obviously will be generated from the fuel, and fuel is the glucose. So this hormone, which we have previously seen, epinephrine, ultimately causes the release of the cortisol, if you remember. Okay, so it can, sorry, not, I'm mistaken. Epinephrine is released because of the low glucose level. So epinephrine has like different effects on that cell, such as physiological effect and metabolic effect. Physiological effects are what? The heart rate, blood pressure, and dilation of the respiratory passages increased, because at this moment we need more oxygen. More oxygen is needed, which will be delivered to all the tissues, specifically to the muscle. And ultimately, what are the metabolic effects done by the epinephrine? That is, glycogen breakdown is increased because we need more glucose to do the work. Glycogen synthesis will be decreased, gluconeogenesis increased, glycolysis increased. So these will like increase the blood glucose concentration, and another one, glycolysis, which will be ultimately generate the ATP. Fatty acid also mobilization is increased. That means we know that it is stored as triacylglycerol that will be broken down, and fatty acid is released, so that fatty acid can be utilized as fuel. Glucagon secretion increased because at that situation the blood sugar level is low, so that will be signaled by glucagon, so the cells then can take the measure by increasing the synthesis of glucose, or they can depend on other or utilize other fuel, fatty acid or amino acid. And insulin secretion decreased. So these two hormones actually reinforce the metabolic activity or metabolic effects of the hormone epinephrine. And the last one is cortisol. So cortisol is actually the hormone. কিভাবে হচ্ছে সেটা তো আমরা দেখেছি, when our central nervous system is getting the information, that means different stresses such as anxiety, fear, pain, hemorrhage, infection, low blood glucose, and starvation for a longer period of time. So in that state, the adrenal gland, which is called the adrenal cortex, is released the hormone cortisol, and cortisol then acts on the target cells, muscle, liver, adipose tissue. And why it is like signaling those? Because at that situation the organism as a whole must control the stress by the normal function of the cells, and when the cell need to do normal functions, the cell must have enough fuel. But cortisol is a slow acting hormone because cortisol is a steroid hormone, and cortisol binds to the receptors present within the nucleus, and then that interact with the regulatory sequences of the gene, and that then like control the transcription, and eventually that will be translated into the protein, which will then go to the cytosol and do its function. So that takes time. And here the particular enzyme, the cortisol actually inducing that is phosphoenol pyruvate carboxykinase, and that enzyme is involved in the gluconeogenesis pathway. So that means here cortisol is like um, informing the cells to get ready to produce enough glucose by gluconeogenesis by the action of the enzyme phosphoenol pyruvate carboxykinase. And ultimately, that glucose will be produced to control the situation, or it can also stimulate muscle protein breakdown, and those amino acids then they will be transported to the liver, and amino acids are utilized to synthesize glucose by gluconeogenesis. So now the glucose, that organism has enough glucose to control the situation. Is it clear? But cortisol, sorry, is a slow acting hormone, and that's why it takes time. Is it clear?
Yes, ma'am.
Yes, ma'am.
Okay, so our very last slide, body mass index, BMI. So that BMI is calculated by uh, dividing. So we have to divide our weight. Weight is measured in kilogram. We have to divide it by our height square. Okay, and that actually is a indication about our health or metabolic state. So if the BMI is below 25, this is normal situation. If it is within 25 and 30, it is the indication of overweight. And if it is more than 30, that indicates obesity. And obesity is actually, this is a severe indication, and which can be life threatening, because obesity is related to uh, uh, um, various diseases such as type 2 diabetes, heart attack, stroke, and cancers. So that's why it is important for us for healthy life. Okay, so that is about our integration of metabolism. Do you have any question? Any question? Anything? And I think I have already informed you about the question pattern. Is it
Yes,
clear to
ma'am.
you?
Yes, ma'am.
Okay, so I said that uh, fatty acid catabolism, amino acid catabolism, from each one, say we have one question, and TCA cycle and oxidative phosphorylation together we will have one question. That is the catabolism part. And then from anabolism, uh, biosynthesis of carbohydrate, one, biosynthesis of lipid, one, and then finally amino acid biosynthesis, nucleotide, and the integration. These three together will be one. And we don't, I'm not sure whether, but one part or like majority part may come from the integration, and the minor part may come from amino acid or nucleotide. Is it clear?
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
Okay, so if you don't have any questions, so we can end here. And also just for your to make it very clear, our exam is on 19th, but if there is any problem on 19th and the Thursday, that is the 21st, if it is a normal day, we will have the exam on that day.
Okay, ma'am.
Okay,
Okay,
ma'am.
then see you. Please study, prepare for the exam sincerely, give your concentration, and it is possible to do good.
Yes, ma'am.
Okay, Allah Hafiz.